Bismillahirrahmanirrahim. Today, inshallah, we'll talk about Chapter 8. The first part of Chapter 8 is about cellular transport. When we were talking about cell structures, we had mentioned different organelles and membranes, etc. So we had alluded to this before. But here, specifically, we're going to focus on how things move about in and out of the cell. It's a very important topic, actually, and it's an important topic even in real life, like in medicine, in taking care of patients. We need to know this. The first important concept is diffusion. So diffusion is a very important concept to understand, and we have already suggested that diffusion is the movement of particles from area of greater concentration to area of a lesser concentration. That's easy enough, and I had suggested before why that is the case. Okay, so diffusion is movement of particles, as you can see, from area of higher concentration to the area of lower concentration. It is a matter of probability. The reason it occurs in this manner is because of probability of these molecules bumping into each other is higher in the area of greater concentration, and until that probability is equal, diffusion will continue to occur. So that's diffusion. There's a slightly different term, and that is osmosis. Osmosis is a specific example of diffusion, because anything can diffuse. Previously we talked about a drop of ink diffusing in a beaker of water, right? So in that case, what was diffusing was the ink. But water can diffuse too. Water can move from the area of higher concentration to the area of lower concentration. That's diffusion of water. Now, so that process is called osmosis. So diffusion of water is called osmosis. The tricky thing about water, which confuses some students, is it's a difficult concept to sometimes appreciate, because most things are dissolved in water. The ink was diffusing in the beaker of water. A drop of blood will diffuse in water. So most things are dissolved in water, right? So most things diffuse in water. It's hard to kind of think, what would water diffuse into? Would water diffuse into a barrel of ink? You know what I'm saying? It's kind of like that. So it's a little trickier in that way, but the concept is the same. All you have to figure out, where is there more water, even if there's more water by 2 or 3 more molecules, so to speak, extra. You follow what I'm saying? So diffusion of water is osmosis. Okay, so which means that water will move from area of higher concentration to area of a lower concentration. So here osmosis is water going from higher concentration to lower concentration. Diffusion, for example, could refer to diffusion of oxygen molecules, carbon dioxide molecules, or any molecules otherwise. So again, just a specific example. Now, there's some terms that we have to understand, okay? The first term is solute. A solute is typically what is dissolved in a solution, okay? So whatever you're talking about, okay, whatever the thing that's dissolved, the dotty things, that's the solute, okay? Solute sometimes can be a salt. It doesn't have to be. It can be a sugar. So whatever is dissolved in water or whatever it is, that's called the solute, okay? So now look at this. On both sides there's water, okay? On both sides there's water. And water molecules in this cartoon picture are represented by these tiny little dots, okay? Now, if you add anything on either side, for example, over here, sodium chloride. Now, the sodium chloride takes up space where water would have been, right? How else are they going to get there? They're not going to be there on top of water. You know what I'm saying? They have to take up space. So, because there is solute over here, there is more water over here, and there is less water over here. In other words, the concentration of water is more over here and less over here. The water will therefore move over this way. Now, another way of saying this is, now it's very easy enough if you think about it that way. Where there is more water molecules, the water will go from the area of more to the area of less. Okay? Another way of saying the same thing is water will move towards the solute, whether the solute be sodium chloride, it is salt, or it be sugar, right? So when I was in high school, I remember to this day, my high school teacher had suggested, salt sucks water. It's almost like it sucks water out. You follow what I'm saying? Wherever salt is, or wherever sugar is, wherever the solute is, wherever the solute is, it's like sucking the water on that side, right? It's not really sucking the water, right? It's water that's doing the movement from area of higher concentration to area of lower concentration. Do you understand? So there are just two different ways of looking at the same thing. What's really happening here is osmosis, water moving from area of higher concentration to area of lower concentration. But another easy way of remembering that is salt sucks. That's what my teacher told me a long time ago. Salt sucks. So solute basically drives, okay? So that's an important concept to understand. Now look at this over here. The same thing, I'm going to elaborate on this tiny bit, okay? So look at this. Now this here represents two compartments, okay? And here are the water molecules, but these blue thingies over here. And on this side, there are solute molecules. In this case, not salt, but rather sugar, okay? Sugar molecules. So now, if you put sugar molecules on this side, now notice, the sugar molecules are pretty big, and we already know from a previous lecture that sugar molecules don't cross membrane. And this happens to be a differentially permeated membrane. Another way of saying that is selective permeability, similar to plasma membrane. So this is just no ordinary membrane. It's a membrane that resembles the plasma membrane in that it allows water to go across, but it does not allow sugar or solute to get across. If that is the case, then the water, as you can also appreciate, will go from left to right because it's going from area of higher concentration to area of lower concentration. Or salt sucks, but in this case we don't have salt. In this case, sugar sucks, so to speak, right? You follow? So wherever solute is, it's going to drive the water to that place. Now, if you understand that, we can build on this. And build on it because the terminology can be tricky, okay? Now, on this side you say there's low sugar concentration. You agree with that? On this side there's high water concentration. We saw that. And on this side there's high sugar concentration and there is low water concentration. Yes? Yeah, we got that. So high solute, salt, sugar, okay? And low water concentration. That's on this side over here. Okay? Now look what happens. The same scenario. Okay? Now this is a selectively permeable membrane. It's not allowing the sugar to go this way. Then the water will go over here until the concentration of water is equal on both sides. You follow? So low sugar concentration, high water, and high sugar concentration until the water concentration is equal on both sides. Okay. So now there are these terms, okay, that you have to know. Hypotonic, hypertonic, okay, and tonicity refers to the amount of water that there exists, okay, compared to the solute. The easiest thing to remember, this refers to solutes, okay? Solutes, not water, because it's easier to think about solids. In fact, okay, I'll give you a real life example, okay? A real life example in just a little bit once you understand this, okay? It can make a difference of life and death, okay? And hypertonic means there's a lots of solutes, like hypersolutes. There's lots of solutes. Okay, so think in terms of solutes. Hypertonic means lots of solutes. Hypotonic means not that much solute. Okay, and remember, salt sucks, or solute sucks, or sugar sucks. So in a hypertonic solute, where there's a lot of solute, the water will move obviously from here to here. Why? Is it because there's more solute there? Nah, really. What? Because there's higher concentration to lower concentration, osmosis. So the water will move from this area to this. So it will go from hypotonic to hypertonic area until there's equilibrium, until the concentration is equal, and until the concentration of the solute is equal on both sides, because that's what's driving the movement. Now, saying the concentration of solutes is both on the same time is basically saying the concentration of water is the same on the both sides, but it's kind of trickier to understand that way. It's easier to think in terms of the solute, okay? But what we're really talking about is water movement based on water concentration. Does that make sense? So now look at this again. When you say a solution is hypertonic, okay, you are referring there's lots of solutes, like lots of solutes. When the solution is hypotonic, okay, there's less water. Okay. You know what happens? The number one infectious disease killer, the number one killer in the world is cholera. Cholera. It's a diarrheal illness. Bacteria, cholera, causes diarrhea. And people get dehydrated very fast and they can die within hours. Okay. The commonest killer infections. Okay. Now what happens when people get dehydrated, they get dehydrated. Okay? Right? They lose their diet, they get dehydrated, and they start drinking water. And it's drinking water that kills them. Why? Because when they lose water, from the diarrhea, they're losing water, they lose a lot of electrolytes and they lose a lot of ions, they lose a lot of solutes with it. So they are losing hypertonic solution in their poop, but they're replacing that with the hypotonic solutions. No. You can't do that. We'll see in just a second. So they're losing hypertonic, they're replacing themselves with pure water, and they die. And this death, in fact, is so preventable. All you have to do is, instead of drinking water, you drink Gatorade, because Gatorade has sugar and electrolytes in it, so to speak. You don't have to drink Gatorade. In fact, a simple mixture could save the lives of millions of people. Sugar, bicarbonate mixture in water, instead of pure water, could save lives of millions of people. So this phenomena, as simple as it seems, is actually very, very important, and it involves life and death situations in parts of the world. Okay? So now look at this. Now, you understand that whole hypertonic. Okay, now watch, okay? Now, we are going to just elaborate on this concept just a little bit more. I think we can handle this, okay? Now, hypertonic means there's a lot of solute. 10% solute, just 10, okay? On this side, there's hypotonic solute, means less solute, 5% solute. So the water will move from here to here because the concentration greater for the water. Until when? Until the concentration of water is the same on both sides. The concentration of solute. Until an equilibrium is established. And only those molecules that are permeable will move. If the solute could move, it would move too. Do you understand? But in this case, it cannot. Does that make sense? Now here, look at this example over here, okay? Sorry. Look at this example. Now, this is supposed to resemble cell membrane. Cell membrane. There's water and there's solute. Now, the solute cannot move across the cell membrane, but the water will. Okay? So this phenomenon occurs in living cells. And this continues until the water is equal on both sides. Okay. Now look, now that you understand this, okay, you can understand there are basically three different scenarios that can occur. Now we talk about living cells, because who cares really what happens in a beaker, right? You know, we really want to know how this affects living things. Okay, biology, after all, is the study of beakers. No, it's the study of living things, right? So there are three particular scenarios that can occur. One is the cell the cell, or maybe it's an amoeba or bacteria, is bathing in a hypotonic solution, meaning there's very little solute on the outside compared to the cell. Hypotonic means compared to the cell, there is less solute on the outside. Now, isotonic, the cell could be bathed in, it could be swimming in, it could be inside it. Isotonic means it is equal on both sides. It's equal on both sides. And then a cell that's bathing in a hypertonic solution means there's more solutes on the outside than there is on the inside. Basically, three scenarios, right? Either they're both same on one side, either there's more on the outside, there's less on the outside. There are three potential scenarios. So these are terms that are very important to understand. Okay, now look. Now each of these scenarios will have different implications. Now this happens. So now we're talking about no ordinary cell. We're talking about a red cell. Okay. Now if a red cell, this is a normal red cell. Now here the red cell has, the cartoon picture, okay, has 3 solutes and the outside has 3 solutes. Now they're both equal. That's the isotonic solutions. The number of solutes outside equals the number of solutes inside. So the net movement of water, the water will still go in and out, nothing stopping it, but the net movement is the same. Does that make sense? But if this red cell is in a hypotonic solution, or there'd be red cell jellyfish or whatever, if it's in a hypotonic solution, that means there is less solute on the outside compared to the cell, then the water will go inside the cell, and the red cell will get chubby, swell, just like somebody's joint swells if they hurt the joint, right? It swells until it bursts. And when the cell bursts, mind you, what happens? You don't never hear it. It's not like a firecracker burst. When the cell bursts, all the contents, the insides of the cell, the red cell doesn't have a whole lot except for protein because it's a special cell. But there's any cell where it bursts, all the nuclei, all the cellular organelles, which outside, right? All you would have like the membrane, right? Like a ghost. You follow? Okay. So if a red cell were in a hypotonic solution, it would swell up and burst. If we were in a hypertonic solution, even though there's a lot of solutes on the outside, hypertonic solution, the cell would just shrink because the solute would just suck the water out of the red cells. You follow? If you were to put the cell in a beaker, for example, over here, right? Now this cell is in an isotonic solution, nothing happens. This cell is in a what? The water is going inside. It's in a hypotonic solution, so it swells. And this cell is in a hypertonic solution, so it shrinks. Okay, so it shrinks. And this happens. If some patient comes, for example, in the medical profession, give a real life example, when people come in with burn victims, right, they lose a lot of water from their skin, right? But if you don't replace them, patients go to the hospital, they get saline, they get IV fluids, right? IV fluids normally they're supposed to be isotonic, same tonicity compared to the blood. If depending on certain circumstances, you have to give different fluids. If you give the wrong fluids to the patient, in fact, the fluids that are hanging that are supposed to help the patient can end up kill the patient because of this disparity or mismatch of the tonicity of the solution and the tonicity of the red cells. Does that make sense? Okay, so this is very important. So another representation, the same thing. A red cell in a hypotonic solution, a red cell in a hypertonic solution is being sucked out, right? And isotonic solution where everybody is happy. So as I said, this does happen. Here's red cells shrinking. Bursting. Now, over here, you can see over here, okay? And you know what this is? These are red cells under a microscope, all of this, okay? These are normal red cells, okay? Normal red cells. Okay, over here, they're in hypertonic. The water being sucked out. They look like shrunken, shriveled prunes. like khajurs. Yeah, they're shrunken. You see, because they're very hypertonic solution. You see this? Going this way though, they're in a hypotonic solution. They're getting chubbier because they're taking water in. Until, what happened to all the red cells over here? They're gone. They burst, see? But you can still see their ghost. Ooh, you see that? They're there. They just exploded. So this does happen in real life. Okay. This can happen to plants too. As we remember, we said that the central vacuole of the plant is very important for maintaining its turgidity, maintaining its strength. When the plant is dehydrated, like in a long summer day, for example, like this, the plant wilts. But the plant wilts. The plant, however, the plant cell has a distinct advantage over a red cell. Yes? And it won't burst. Because, unlike the red cell, plant has a cell wall. The cell wall prevents the plant cell from bursting or having serious damage. So when you do give the plant the water, the plant will become healthy again, will become strong, stand up tall. You follow? So the plant is protected against these changes because of the cell wall. Okay, so here's a normal plant. Here's a wilted plant because of this. The central vacuole is shrunken because it's dehydrated. Alright, so far so good. What's going on in this picture? I wonder. So this is osmosis. Okay, same thing as review. Water will move from the area of higher concentration to the area of lower concentration. Diffusion of water is osmosis. And it occurs simply as a matter of probability. You follow? And this here is trying to show that each particular molecule is just doing its own thing. It's not like it's got a brain. It's simply a matter of colliding into other molecules. Okay, so that is the first part of this talk. We're talking here about movement of stuff. The first thing we talked about is the movement of water across the cell membrane because it's the one very, very critical thing that happens all the time to cells. The second part, inshallah, will be about talking about transport across the membrane of other things, such as proteins, ions, and molecules like glucose, etc. after a brief pause. Until then, as-salatu was-salamu ala rasulillah. Assalamu alaikum.